All right. So in this tutorial, I'm going to be going through how to use boosted regression trees to train a model using PACE data. I'll be using the RRS data, all wavelengths, to train a model to predict surface chlorophyll. And at the end, what we'll be doing is trying to come up with, are we not trying to, coming up with these predictions like this. Here I am for one day. It's fed in the PACE data for that day and then made a prediction of chlorophyll from this boosted regression tree model. And it's comparing here to what the PACE chlorophyll model is like. You can see there's quite a bit of similarity. Okay, so when we are fitting a model, it's not just boosted regression tree. It's kind of anytime we're fitting a prediction model, it's going to be kind of a similar framework. So first, we need to come up with some training data. So we decide what we're trying to predict. And so we'll need observations of the thing we're trying to predict. And then we need the explanatory variables. So our end goal that we need to fit to this model that we're going to train will be a table that looks like this. Again, this isn't specific to boosted regression trees. It's kind of anytime we are preparing or for most models that we're preparing for training. We get a table like this. These are observations and then our predictor variables. So in this case, what I'm training on is I'm going to use data from Bio-Argo buoys, and I'm using this data that they are collecting throughout the ocean. I'm going to show you that in just a moment. And this Y here are logged surface chlorophyll from this Bio-Argo. So I've got all these buoys all around the world. They're getting chlorophyll. I've got that. And then I have the date that that data came from. This is showing 2020, but yeah, I'm training on data after PACE, so I'm training from 2024 onwards. And then here I'm only showing two RRS for two different wavelengths, but I actually used all of them. So I'm doing a matchup step where I'm matching up, this is the buoy information, and I'm getting the PACE data for that point. And then also I'll explain why I add solar hour. Okay, so let's first back up and I'll tell you a little bit about Bio-Argo data. Okay. Okay, so Bio-Argo data. Let's see, let's go. You can click on that to open it in the hub. I myself am going to open it over here. Argo P. There we go. Okay. Okay. So Bio-Argo, what is Bio-Argo? Well, so Bio-Argo are all these floats. They're autonomous profiling instruments that drift around in the ocean. They are over deep water throughout the ocean. And what they do is they'll descend down to some depth, kind of deep. And then as they come back to the surface, they will collect data. And then when they get up to the surface, they send that data to satellites. And this data is made publicly available and provides a lot of great information about what's happening in the ocean. So to use that data in Python, we can use this package called ArgoPy. And do I have ArgoPy installed? I kind of believe I do. Let's go ahead and run it here. I don't. Okay, so we can go ahead and just install, just for this session, run this code here. and that will install the package. It won't be there when you start up the server again, but it'll be there for while you're kind of doing this. So this shows you how with ArgoPy you can get the data. You have to specify the type of instrument, which is the Bio-Argo. So this is the one that will have your chlorophyll. We are getting this from ERDAP servers, and then we are getting chlorophyll and pressure. Pressure is what gives us how deep the buoy was. So, right, we need to know depth because we need to know where along here it was collecting that data, and it reports that as the pressure. So let's go ahead and do that. Takes a moment. Going out there, getting it. These can be really big files. So there we go. It got it, queried it. And next, we can turn that to X-array. So this gets us our X-array data set. So we have all the tools we've been working with during this week. Going to take a moment. It's kind of big. Oh, come on. It's really taking a long time. Okay, here we go. Great. And so it got that data. It did take a little while, but we got it. And now we can take a look at what's in here. So dimensions, oops, dimensions. So I don't need this. Okay. So each of these are all the points that the buoy was recording for this mission, and this is the platform, but the cycle number is when it was going down and then going up. So when you get this data, it's a whole lot of different going up, going down. And so you want to get the data from each cycle separately. Okay. So how many, so we have like 9,000 points here. And let's go ahead and plot the data that we got here. So what you see here is I took the lat long from the data and I separated it by cycle, because remember, these are each cycle. So we want to see how many times it's going and doing one of these sampling from way down low up to the surface. And I specified it in this box, and you can see got some data from just kind of around off the east coast of the United States. And one thing to keep in mind here, the Bio-Argo data is, it goes down to really deep depths. So obviously it's not going to be close into the coast. Okay. So Bio-Argo is data that's in the deeper ocean. And then we can plot this. Area code. Thank God for code assistance nowadays. So much easier to write code like this. It's like 100 times faster. And what we can see here is this is one cycle. And here is the measurement of chlorophyll, milligrams per meters cubed. And you see here's our log 10 depth. And this is the chlorophyll. And this is the surface up here. And then this is about 100 meters down. And yes, Shelley, the tutorials will they're permanent up there for a long time, as long as we can keep them. Yeah, so this shows something that's really interesting that we see out in the open ocean, is that the peak chlorophyll might not be up at the surface. We get these peaks of chlorophyll that can be occurring kind of deep. And one of the things that happens in some parts of the ocean is that the upper surface is nutrient poor, and the nutrients are lower down. So the plankton needs to be down by the nutrients to grow, but they can't be so deep where the nutrients are really strong because there's no water down, no light down there. Like we just heard Ryan talking about, light attenuates. So they're kind of like trying to weigh these two things. I want food, but I need light too. And so that's how you can get these peaks that are not necessarily at the surface. So we see this. This is really common that we'll see these peaks below the surface. And so what I did, and what we'll see in this next tutorial with the boosted regression tree, is I went through all the Bio-Argo data, and I was looking for the average chlorophyll in the surface. So what I'll show afterwards in kind of my sciencey part is estimating the full profile down to 200 meters, which is kind of as deep as these peaks tend to go. So I'm going to show you work estimating this profile that you see, but for the tutorial, we're just going to focus on the upper surface so I can compare it to other chlorophyll products. And how do I do that? So basically, I needed to create a function that would go get a profile, look at the number of points that are in the upper 10 meters, and take the average. So I'm not going to go through that code here, but this just chugs through all of them and gave me a nice big data set. I think over 5,000 points. We'll see in the tutorial. I forget exactly how big it was, but it's plenty big enough to train a boosted regression tree, even after matching up to PACE, where I lost a bunch of points because there was missing values, as you all have seen. Okay, so this data set that I created, it's one of the data sets we have available in the fish-based data sets. You are welcome to load that up. You can load that up like this, and you can play with it too. All right, so now let's go to the next tutorial, which is before I leave that, I'll just note here, I also have a similar tutorial on getting chlorophyll data at different depths from this mooring, this ocean observing, what is it? Yes, the Ocean Observatories Initiative. They have a lot of moorings out in the ocean collecting a lot of great data, and I show you how to get that data also. This tutorial, if you are doing like kind of heavy duty matchups with lots and lots of points. So for this one, I don't know, I think I had many thousands of points to do matchups, which can get computationally hairy. In this tutorial, you will see how I do that using this kind of batches so things can run for a long time. Okay, but let's not do that. Let's get into those details. Let's hop over and now go to this boosted regression tree. So if you want to run this, I did try to make it so it would hopefully run on a 2 GB RAM. Maybe, but it should run on a 4. So if you opened up a 4 for Ryan's tutorial, this should also work fine on a 4 GB RAM. Okay, so reminder, if you haven't done that before, you're going here, you're going control panel, you'll stop it, and then restart it and pick a larger instance. Okay, so let's go. I'm going to go into the tutorials. Okay. So this is the one that is on the website. And So let's walk through the steps for fitting a boosted regression tree. So a boosted regression tree is a model where it's it's explaining the data with all your parameters, and it's creating these kind of trees where it'll take your parameter, like one explanatory variable, and it'll like break it, like if it's greater than this or if it's less than that, and it will make kind of a break. That's how it's explaining, a higher, lower, and then it breaks it again. And so it keeps like breaking your uh, your explanatory variables in kind of these cuts. And one of the nice things is that it's able to model nonlinear relationships, and also in um, the kind of your like established packages for fitting boosted regression trees. They're kind of designed, they are designed to not overfit the data. So they have kind of guard rails on it, and so that you can have like lots of variables, and it's okay if they're all correlating. It handles the correlation in the variables because it explains with one variable, and then it explains the residuals that that variable doesn't explain. So they're tend to be robust. I mean, if you're using like an established package, like I'm going to be showing you today. I find the package I'm going to show you today really nice for like prototyping ideas. It's a really fast way to see, hey, is there signal in the data that can explain what I'm trying to predict? All right, so basically what I'm going to be doing here is I'm going to be trying to explain that surface chlorophyll that I got from the that I got from the Bio-Argo data. So that's my real data that I collected, my in situ data. And then for each of those points, I'm going to get all the RRS wavelengths. So all, what is it, 172 of them. And that's going to be how I'm going to try to predict chlorophyll. I will note I'm also adding on solar hour, and that is because I know something about the instruments on these buoys, and it's very well known that the buoys are affected by how overhead the sun is. So that's a known thing that you need to correct for. So instead of applying any particular correction, I'm kind of telling the model to say, hey, find that correction for me, which is what it's doing by adding solar hour in there. All right. So let's see. Okay. So here's the overall steps when you want to fit a boosted regression tree. So you're going to need to create this data frame like that. And then you'll fit the model and you'll make predictions. So in our model, like I said, I've got all the RRS, and I've got the solar hour, and then I have this chlorophyll I'm predicting. Let's see. So just to mention about NAs, I do want to remove those from my data frame before I'm fitting my model. However, I think the boosted regression functions tend to filter are robust to that. I mention that because I also do a lot of convolutional networks, and we have to spend a lot of time making sure there are no NAs anywhere. The other thing that I will note is that for your response variable, in this case, the chlorophyll, I do want that to not be like skewed. So I'm taking the log so that it becomes kind of like central, but I don't have to scale or transform my response variables. That's another really nice thing about the nature of boosted regression tree algorithms is they are fairly robust to the scaling and the shape of your response variables. So response variables in my case are these RRS wavelength. Okay, so I don't need to run that because I'm on the Jupyter Hub. I am going to restart my shut down all kernels. I got 3 running. Let's just make sure I have plenty of data. Okay, great. All right, so load up this. Okay. I have created some custom functions so that I didn't have to have all of them in the notebook. And in Python, you can load up a file with defined functions, and then you can use them in your notebook. Just like how we say like import Xarray as XR, and then we can use all the Xarray functions. Same for functions that you write yourself. So you don't have to have all your functions in your notebooks. You just keep the like when I'm working, I have one file that has the functions I'm using. So here I have all the functions in this. Most of these functions I'm not using now for the notebook just to like kind of show everything. If you go messing around in the notebooks where I'm applying this, when I talk about the science part, you'll see I have kind of everything in this in my Python files that I call. So this is how I'm going to call this in, and I am actually calling it in from GitHub. I did it this way so all of you can run it. So you're sourcing a file that's on GitHub. So we'll go ahead and run that. So I've sourced in my own functions that I had on GitHub. And now we're going to load in the Bio-Argo training data. When I started, I showed you how I created that data by processing the Bio-Argo. So this is the data that I'm reading in. And in that data, I already did the pace matchup. So I'm not covering that part. I got all those Bio-Argo data with the time lat long, and then I went through, took about 5 hours getting all the pace data and added that to the table. So that's there also. So I got that. And then I'm going to add on the solar time. So this is a function that I have that looks at the lat long and looks at the timestamp, and it tells me what is the time relative to when the sun is vertical. So that's the solar time there. These other things, I'm not going to be using those, but I will note here that when we're doing boosted regression trees like I am on a globe, sometimes we want to have a different model for different parts of the globe. And the if you just use lat long in your model, it wouldn't work quite as well. You use something called spherical coordinates, which are an XYZ, and it's got a unique point for each part of the globe. This will behave better. So I just point this out because it is really common that you might want to include that. Like if you want, hey, I wanted to recognize that I'm in the Arabian Sea versus the North Atlantic, and maybe the way the model should behave should be different in those regions. If you thought that was really important, you could include the spherical coordinates. I also, if you go poking around in that Python file, you'll also see I have a function for distance from coast, because for some of the things I do, I want to know how far I am for a coast. I think that is affecting my predictions. Okay. So you can add all those things on. But right now, I'm just going to be using the solar time. All right. So this is what I end up with. At the end, I have about 2,700 points to use to train my boosted regression tree model. 233 columns because I've got that, what I'm trying to predict, and then all those wavelengths. And let's just take a look at where these samples are. So here's where my samples are. They are over from 2024 February to, I think I finished this in November 2025. And you can see we've got, more coverage in the Atlantic, but we have points all over the globe, except, not real close to, the poles here. All right. And yeah, keep in mind what I'm going to show you is I'm just training off the Bio-Argo data. And so remember, I am training more on open ocean data. Right, so now we've got our data frame. And now you can fit a boosted regression tree. And what I'm going to use the sklearn package, and it has a function to do our training and test split. So we're going to take our data frame and we're going to use some of the rows for training and other rows for testing. So we're going to train on one part and then we'll test it. So we'll look at performance for data that we never saw before. I am not going to do any kind of fancy other type of splitting. I will note, like sometimes like if you have seasonal data, time series data, you often will use, fancy types of splits. I leave that for you to research. So I'm just doing a basic split. So what do I need to do? I need to tell it here's my response variable. So I'm saying, hey, use that. And then here are my variables that are going to be the things I'm trying to predict with. So these RSS columns. I add on an extra one, which is the solar hour. So right here, what I'm doing is I'm saying, hey, this is my training data set. It has time, lat, long, the predictor, and then what I'm trying to predict and what I'm trying to predict with, which are these two things here. I do a little renaming. I do take the log of my response because I want it to be normal-ish. And there we go. So I pass this function wants those, data frames. And, well, you can do, if you look up help, just look up the documentation for this, it'll tell you exactly, what it needs. But you can kind of see basically what's going on here. It needs this one. The X is going to be your, what you're using to predict. So the X is what you're using to predict, and then your Y is your target. So it takes your Y and takes the things you're doing to predict. It takes how much to hold out for testing. And let's see here. We'll just run that. Okay. So there we go. A little fiddly to get it right, but once you do it a few times, it's not terrible. So here's the X. You can see it just has my predictors in it. It's clean, just has that. And the Y is just the thing that I'm trying to model, what I'm trying to predict. So now I have what I need to fit a boosted regression tree. I'm going to use this function from sklearn, and it wants, you can see right here, you use that, you specify a few things. I just use like standard kind of defaults. Honestly, I think I just asked, ChatGPT, hey, what's kind of standard? And that was kind of good enough for this tutorial. And then you use the fit function and you give it what you're training, the X train and the Y train. And that's it. Um, Thankfully, like I said, like I mentioned, I used, the coding helpers. They know sklearn really well. There's been so much material put out there, so they know it great, which is really nice. It'll write you really clean code for fitting boosted regression trees. All you got to do is tell it what your data frame looks like. Okay, so now I've got my model. And now I can make some predictions. So here's my predictions. I say they're my model. I predict. And then I'm going to show residuals first. So how well the model fit the data it was training on. And then I'll do the same thing for the test data. So this is my residuals. So I'm going to get prediction from the training data, and then the residuals are the difference between this Y train is the truth, and this is the prediction. So the difference between those is the residuals. Let's see how it did. So there's the residuals. We're going to make a little map here. Oh, this is a little helper function. Make the map. All right. So there's the residuals. It's on the log scale. And yeah, all right. Okay, let's compare to the test data. For this, I'm going to show the R squared. Hey, there we go. So at least I was able to fit the data, R squared of 0.73. Okay, yeah, that's way better than random. And let's, again, we're still looking at how well we fit our own data. And I mean, fit the training data. And yeah, here you go. Huh, that is nice. Okay. So what that tells me is, yeah, these spectral shapes do predict chlorophyll. I mean, surprise, surprise, we know that they can, but I was able to with a pretty just sort of like throwing at it a booster regression tree. I didn't, throw any knowledge about the physics or anything or any known like shapes that it should be. I just said, hey, figure out what would work, and it did. This is how well it is fitting the data it has not seen before. And yeah, you get some high numbers, but generally it's kind of hanging out around here. So, hey, doing pretty well. Okay, so next, what I would want to do is say, okay, so it seems to me that the RRS for all the wavelengths, this model that I fit, it's way better than a cloud. So I'm going to make some predictions. So now I can go to a day, whole day, take all the paste data, and then use my model to make a prediction for that. So at this point, the data starts getting a bit hairier, a bit longer. The basic idea here is that, so I'll just describe kind of what's happening. So let's see here. Let's go down. Okay. Okay, this shows chlorophyll, but imagine this is RRS, and it's got a whole stack of those. And it is going to take that RRS for this scene here. This is what I'm going to be predicting. And it's now going to create this big old pandas data frame with for each pixel, it's got all the RRS values. And then it's going to tack on, I'm going to give it also solar hour or tell it what solar hour to use. And now it's going to make a prediction for that pixel. And so now I'm going to get a data frame with a prediction for each pixel, and then I'll have to reassemble the thing into a map. So it looks like it's got lat long. that's kind of what all this carry code is doing. I did try to write the code so that if you wanted to use it, fingers crossed, you can just use it. Hopefully or with minimal changes. So I am running that. I ran these cells to define these. I did that very quick. Let's do that a little bit slower. So I'm going to get that function. This is going to get that the RRS netcdf file, and it's going to grab all the values and make it into that pandas data frame. And then it's going to take that data frame and it's going to make predictions and then spit out a map. That's what the second function does. All right, so now I've created those functions. So first thing is I need to get a netcdf for a particular day. So here we go. I also got for chlorophyll just because I want to compare that. And I pick a day, so I'm going to do July 8th. I should have used that OC view and picked a day with no clouds, but I didn't. I just kind of like poked around until I found something that had a fair bit of data. So now what I'm doing is I'm going and getting the netcdfs, the RRS and chlorophyll for that day. I don't use the chlorophyll in any way for training or fitting the model. I'm just going to compare to my predictions, just kind of as a sanity check. Okay, so now I have that, I got that data set for that day. Let's take a look what I just got. So I have that, okay, for the whole globe here. And now I'm going to grab a bit of that for this North Atlantic. I picked a really small box. I could do it globally, but so the way I wrote this tutorial, I wrote it to have the code simple. I wanted the code to be simple, but that meant that I had to write it that when it makes a prediction, it has to create a pandas data frame that it loads into memory. So what that means is like all those really nice things where I said, oh, I talked to you about like chunking and everything. Because of the way I'm doing it, I have to load the whole thing in memory. And so I have to predict for a fairly small box. So that's what's going on here. I got a small box, make that prediction. So if you were to make this a lot bigger, it will blow up. Not there, that part won't blow up. This is the part that's about to blow up. So we can watch the memory here. So now what it's doing is it's using all those RRS and it's making a new prediction. There we go. And we use the little function I wrote that takes that prediction and reassembles it into a map. And there we go. So this is my prediction. This is compared to the PACE chlorophyll product. Now, I don't expect these to be the same at all. I mean, I would hope they're similar, which they are. But first off, they are developed differently. And I have just trained on data that was only in kind of the more open ocean. So it's no surprise that the model has never seen coastal data, that it wouldn't do well in the coastal environments. It's doing a lot better out here. You can see the colors are more similar versus like right along the coast. As I get more coastal data, hopefully it'll do better. Okay. We can now, I'm going to make a little plot of just kind of to reiterate that this is the PACE versus the boosted regression tree predictions. And so this kind of bias where the boosted regression tree is it's always a little bit lower is expected. Won't go into that. It has to do with what the models are trained on different things. But they should be correlated, which they are. This is out in the open ocean. And then you can see that when we're in the coastal area, it doesn't do so well. And that again is expected. This model has never seen coastal environments or really high values, so we wouldn't expect it to be able to predict there. All right, so that concludes the demo part of how to do a boosted regression tree. I will next show a kind of what I am doing with this data. But I want to stop here and see if you have questions that you want me to address before I move on. No questions? All right. All right, Brian. Yeah. Booster regression trees are fun and they're a little daunting the first time you do one, but jump in and, once you have a data frame with what you want to predict and things you want to predict with, after that, it's relatively easy because now we have chatbots. You just tell it what your data frame looks like and say, hey, I want to fit a boosted regression tree, and bam, you're going to have probably working code in 2 seconds. Okay, so next I want to talk about kind of how I'm actually using this. And this is very much work in progress. So this is still very drafty. Okay, so from Ryan's talk, we hopefully you understand that when we're getting these observations of the ocean from the ocean color sensors, it's not like it only sees the surface of the ocean. The light is going down into the ocean, and some of the light reflects off deeper in the ocean. So that suggests that maybe we could see chlorophyll that's below the surface. Maybe there is information in those. We've seen all those like spectral shapes. Maybe there's information in the wiggles about what's happening below the surface. And we can imagine that might be true because of how, Ryan showed us that the light is shifting. So even though like we might have a big signal from the surface in one part of that, perhaps there's a signal of what's happening lower down in some other part of the spectrum because it's kind of shifting of what's returning as we go down. So that's kind of the idea that maybe this could work. So why would we care about that? Well, I showed you that there's definitely a lot of cases where the peak chlorophyll is not happening right at the surface. And fish, other creatures that depend on plankton are targeting that those blooms that may be happening, or those peaks that are happening in different parts of the ocean. So that where that peak food is determining the three-dimensional habitat that fish experience or whatever else is eating them. Also, it's important when we're trying to study the carbon cycle, because if we are only getting the estimate for the top surface and we're not accounting for what's happening below the surface, we could be underestimating how much chlorophyll there is. Okay. So the goal of this work was to, I'm going to just jump ahead a few here. Okay. The goal of this was to try to use the hyperspectral data to predict the chlorophyll below the surface. And so the blue lines are the observed, and this is coming both from the Bio-Argo and also mooring data examples. And what you can see here is, here's the observed, and you've got the peak there. And kind of spoiler alert, there is a signal in that spectral data. So this model doesn't have any environmental parameters yet. May add those. Certainly for coastal, I think I'll need to add them, but this is more focusing on the open ocean, and just with that full spectrum, with all the wavelengths, it is able to get a signal for the lower depths. And the way this model worked is I showed you fitting the model for the top bend. This is a model where each point here is a depth bend. So this is the first 10 meters, second 10 meters, and all the way down to 200 meters down. And a separate boosted regression tree was fit to for each depth. Variety of reasons that I needed to do that, but also part of it is kind of the logic of it that, I don't know what the signal might be, how that spectrum might be responding, or what information in the spectrum will tell me about these really deep layers. Nonetheless, this is indication that, yeah, it is able to see down below the surface, at least in these more open ocean environments where the water is a little clearer, a lot clearer. Okay, so what can then be done with that? Well, this is a product that I've been developing where I fit it to all the PACE data and create these global maps. So right now, I do have these global maps in cloud optimized data. And so you can look at the whole globe, and it has both the chlorophyll at each depth and then where the peak occurs. And you can see some kind of interesting seasonal dynamics looking at that. Now that that data set is available, that model is trained, and I can now query the whole model, can get things like this. This is looking at where the peak occurs. And so it's kind of opposite here, but this is the surface, and this is deep, and this is for this box right here. And you can see how where the peak is occurring has a definite seasonal cycle to it. So it's up near the surface in this time of the year, and then during the winter, the peak chlorophyll is deeper. Okay. So the general approach is what I showed you. I showed you the example for one depth. This is repeating it for all those depths. And like I said, I've been using that Bio-Argo data, so it gets that profiles, and then using, do I have this here? Ocean Observing Initiative. The next step is to get a lot more coastal data, because right now I don't have a lot of coastal data. All right, I'm going to stop there. If you want to play around with this data set, it is publicly available. I have notebooks showing you how to play around with it, keeping in mind, though, it hasn't been trained on any coastal data yet. This is very much just testing out ideas, and it's still in very much the draft stage. All right.
I had a quick question. Could you